So in the last slide, we saw the phenomena of antigenic drift and shift. And once more repeating, antigenic drift is the rapid modification of viral antigens through random mutation, while the antigenic shift is the ability of the influenza virus to change its outer surface antigen, which can be done due to the fact that the influenza virus genome exists as 8 separate single-stranded RNA segments, each of which encodes a single viral protein. Now, this was the case of, as I was talking about, the H1N1 pandemic. If you look into the previous history of the pandemics of influenza virus, there was a widespread outbreak in 1918 with the H1N1 subtype, in 1957 with the H2N2 subtype, and in 1968 was the H3N2 subtype. In 2009, it was the H1N1 subtype. Just for note and information, clinical immunologists define a particular antigenic shift of an influenza virus by the identity of its heme agglutinin and neuraminidase molecules, since it is the presence or absence of B cell memory to these surface glycoproteins that influences the production of neutralizing antibodies. The new strains that results from antigenic shift are often referred to as the influenza virus subtypes. So this was the story of influenza virus, because right now we are moving through a history of COVID. And now, coming to another important aspect of T cell activation. If you remember that we just studied how the T helper cells and the T cytotoxic cells are activated via signal 1, signal 2, and signal 3. So it is very evident that for all the 3 signals to be perceived by a T lymphocytes, the engagement of the receptors becomes very vital. This picture of the engagement of the receptors, as you can see, this is known as the serial engagement model of T cell activation. So this is an antigen presenting cell, and this is a T cell receptor. The engagement of the T cell receptor by the peptide MHC leads to phosphorylation of the CD3 chains and recruitment of membrane rafts. So, recruitment of membrane rafts. The membrane rafts contain many of the signaling molecules required for the transduction of the TCR signal. So, I'm not getting into the details of the TCR signal. The peptide MHC complex, which when activates a set of a T cell receptor, this receptor is activated because the signal has started moving downstream. You can see from the picture the peptide MHC complex now disengages from the first now triggered TCR and engages a fresh resting TCR. The process of CD3 phosphorylation, RAF recruitment, and signal initiation is repeated as the peptide MHC molecule moves from TCR to TCR. So you can see it is the peptide, this is the same PMHC molecule that is capable of simultaneously activating one after another the T cell receptors. So by the same peptide MHC molecule, in this particular picture, we are seeing that a set of 3 TCRs have been activated and they tend to aggregate. Now, if you remember that we have studied the supramolecular activation complex. Now, in the case of supramolecular activation complex, the interface or the contact zone between the T cell and the APC where the rafts are stabilized and the triggered T cell receptor accumulate is called the immunological synapse or supramolecular activation complex. So the immunological synapse actually it was the initiation of the immunological synapse that began here, and as the PMAC complex engages, activates, disengages, again engages, activates, disengages, goes on doing this, a host of T cell receptors are being activated and then they tend to aggregate. They tend to aggregate at certain points. In this picture, as you can see, the T cell first scans the surface of the antigen presenting cell to determine if its TCR recognizes the peptide MHC present on the antigen surface. If sufficient number of TCRs are strongly engaged, the cytoskeleton of the T cell, reorganized to generate clusters of signaling molecules in the site of TCR PMHC interactions. You can see clusters of signaling molecules are being generated. So this is scanning, TCR engagement, and as you can see, TCR aggregation. These clusters eventually coalesce, they come together into the central SMAC, which contains triggered TCR and PMAC complexes, and the membrane rafts with the signaling molecules. So as you can see, when there is a aggregation of the TCR, the receptors coalesce to form a central SMAC. So we have a central SMAC. This is on the surface of T cells and this is on the surface of the dendritic cells. So in the dendritic cells at the center we have the PMHC and the B7 molecules. For the TCR we have the the T lymphocytes, we have the TCR CD4 oblique 8 and CD28 molecules. The C-SMAC is surrounded by the inner peripheral SMAC. So the central SMAC is now surrounded by inner peripheral SMAC and the outer peripheral SMAC, or in some cases it is called the distal SMAC. Now the inner peripheral SMAC, so this is the inner peripheral SMAC in the T cell. The inner peripheral SMAC is dominated by binding pairs of CD48, that is LFA3, and CD2. The adhesion molecules that first facilitated the T cell APC interaction become concentrated in the outer surface. You can see the actin molecules, they have been concentrated on the outer surface. So the adhesion molecules have now concentrated on the outer surface of the peripheral SMAC. Triggered TCRs that have completed, so these were triggered TCRs. The triggered TCRs, those that have completed their participation in the signaling. So those who have completed their participation in the signaling are then internalized, and the central are then internalized, and the SMAC structure slowly dissipates before the T cell disengages from the antigen presenting cell to commence proliferation and effector. So as you can see, once the T cell signaling activation has been done at the ligand receptor level, the T cell starts gradually disengaging from the antigen presenting cell, and this T cell is now an activated T cell which will now differentiate and proliferate.